information, sending out a response. And every nerve cell is broken down into three different parts. The part sizes or relative proportions may differ from cell to cell, depending where that cell is found, but they all have these three parts. The primary part is the cell body. This is the primary central portion of the nerve cell. It contains the cell nucleus. It's going to contain many of the organelles as well. You then also have a section that are called the dendrites. These are extremely branched sections of the nerve cell. Usually they're relatively short, but they break off into multiple branches. And then you have the axon. The axon is usually a single long sheath, a long extension that is part of the cytoplasm, and the axon is what actually carries the nerve signal away from the cell body. The dendrites receive the nerve signal, it then is processed and is sent along through the axon to the next nerve cell. So there are the different kinds of nerve cells that you might have include things like motor neurons, sensory neurons, and interneurons. And we'll go into more detail about this when we go talk about the nervous system, which is the next chapter. But each of these nerve cells, each of these neurons is responsible for maintaining a different aspect of the nervous system, part of the nervous structure. Now, the nervous system itself is broken down into two components. You have one that's referred to as the central nervous system. Anyone know what this is? The brain and the spinal cord. So every time we refer to the central nervous system, we're literally just talking about the central portion of the nervous system. It's the central portion where signals are sent and where they're processed. This is responsible for integration of all input. So every sensory information that you receive is sent to this central nervous system. It's not always sent to the brain. Some input is received and integrated in the spinal cord. But there's this integration that takes place. There's an interpretation of what the signals mean, and then there's a signal that's oftentimes going to be sent out from the central nervous system. The other nervous system is the peripheral nervous system. If one is central, the other is in the periphery, on the outside. So these are the nerves and the ganglia, the collections of the cell bodies that are outside the central nervous system, that are outside the spinal cord and the brain. These are the ones that are going to be actually receiving the sensory input. Every sense that you have is connected to a nerve cell, and it's going to be sending this input and receiving this input. It's going to be the ones that are receiving the signals from the central nervous system in terms of a response. So the peripheral nervous system is just this whole collection of neurons and ganglia, the cells outside the central nervous system. So they're going to be communicating by sending signals to the central nervous system and receiving signals from the central nervous system. The central nervous system is the coordinating hub. The peripheral nervous system is the outer regions. So if you sort of look at the nerve tissue, we have these three different types: sensory neurons. These are going to be ones that are actually attached to some sort of receptor, some sort of sensory receptor that is receiving information. Where is the sensory neuron pretty much going to be found? In the central nervous system or the peripheral nervous system? In the peripheral nervous system. That's where the input is received. Every time you taste something, see something, hear something, feel something, every time there's an internal sensor in your body that's receiving information about oxygen levels in the blood, glucose levels in the blood, food that's being digested, those are all part of the sensory nerves. They're all in the peripheral nervous system. You have a motor neuron. Motor neurons are typically going to be found partially attached to the brain and spinal cord, but they're going to be sending signals to the motor aspects, to the motor muscle cells, to the glands that are going to initiate a response to a signal. And then in between these two, you have the interneurons. These are usually going to be found in between the sensory, the motor neurons. These are what the central nervous system is going to be comprised of. There are going to be interneurons that are sometimes out of the central nervous system, sort of connecting this process, so you have a continuous chain of information that can be sent. But while these other two are connected to other tissue types on one side, either some sort of sensory system or some sort of muscular or gland system, The interneurons are always going to be connected to neurons on both sides. They're receiving input from one neural cell, from one nerve cell. They're sending that input to another nerve cell. Now, when we think about the animal body, everything that is regulated, everything that is controlled by the animal body, sort of is a result of coordination of multiple organ systems. In some cases, there are individual organ systems that work independently, but most things that occur are multiple organ systems that are sort of working together in order to make a functioning unit. Now, when we were talking about organ systems, we started off with cells. That's the simple cell. We were just talking about tissues. Tissues then group together based on function to form the organs. And then the organs are grouped together into these organ systems. And so the organ systems are sort of this functional unit that serves a specific purpose within the animal body. So, for example, when animal bodies need to sort of have this idea of communication, receiving sensory input, integration of signals, there are going to be three organ systems that are responsible for coordinating all of that information. You have the nervous system is going to be involved in that. You have the sensory system that is separate from the nervous system, and you have the endocrine system that is going to be responsible for that as well. So anytime we're talking about cellular or organismal communication at an organismal level, not talking to each other, but communication within the body, integration of different systems and so forth, those are the three systems that are involved. In terms of support and movement, you have the musculoskeletal system. It's actually comprised of two interrelated organ systems. You have the muscular system and the skeletal system. Why do we sometimes refer to it as musculoskeletal? Because they're directly connected to each other. The muscles are connected physically to the bones. So they are technically two different systems. They're oftentimes grouped together. Regulation and maintenance of the body itself. There are four organ systems that are responsible for maintaining body chemistry, body function, and so forth. These are the digestive system, the circulatory system, the respiratory system, and the urinary system. So they're sort of just going to maintain what's going on inside the body. You have a defense system. The body has to defend itself. It's always under attack by pathogens. Every time you touch something, there are bacteria that are going to be trying to get into your body. I'm going to make everyone here a germaphobe. But your body needs to continually be defending itself, protecting itself from invading pathogens that want to invade the body. So we have a defense system. These are involved the integumentary system. That's the physical barrier or the skin that protects the body, but it also involves the immune system. We have reproduction and development that needs to take place. You could say that that is the goal of all living things, to develop and ultimately reproduce, pass along your genes. So this is enables for the continuity of life to continue from one generation to next. So in the females, you also have the reproductive system that needs to be responsible for nurturing the embryo, nurturing the fetus. Especially in mammals, where we have this placental mammal situation. So those are going to be organ systems as well. Now, when we have the body itself, we have all these organ systems. They're all working together, hopefully. They're all trying to sort of maintain the status quo. This is the idea of homeostasis. As animals have evolved, as we've seen the development of animals, we talked about the evolutionary history of animals. We've seen an increase in specialization of the body cavity, of the body types, of the body structures. Specialization allows for more efficiency. If you have to be doing everything, you can't be very good at everything. If you can be specialized in one thing, you can get to be much better in that. Now, for cells to function at sort of this ideal efficiency, this optimal efficiency, they have to be able to communicate, but they also have to be able to respond to internal and external conditions. There's an ideal situation, an ideal condition in which cells are going to work the best. It has to not just do with temperature, it has to do with pH, it has to do with the amount of oxygen that's being provided, it has to do with blood sugar that's being provided. Every single factor that the cell requires for the ideal chemistry needs to be regulated. And so the bodies need to If it gets too out of whack, the body is not going to function properly. It's not going to function as well. And so the idea of homeostasis is simply how the body maintains this internal chemistry. So this is a dynamic process. Things are always changing. All you have to do is walk outside for a second and you know the temperature is changing. The body's always worried about losing water. Chemical balances are always changing. Electrical balances are always changing. Oxygen, sugar, these are always being used up. So you need to continually bring more in. So this idea, the body is a continually working, continually changing system to sort of try and maintain this equilibrium, this internal chemical balance that is required for the processes of life. So we have a quick animation. Here, sort of going over what homeostasis is, and then we'll start talking about it in a little bit more detail.
All animals must minimize variations in such internal conditions as body temperature, blood pressure, blood pH, level of hydration, and blood glucose concentration, even though the external environment is constantly changing. Some animals are more tolerant of fluctuations than others, but all must maintain a stable internal environment. Animals have evolved various mechanisms to achieve this homeostasis, but all homeostatic mechanisms operate in the same general way. As an example of homeostatic control, we will use body temperature in an endotherm, an animal that uses heat from metabolism to regulate its body temperature. The thermometer on the right shows a mammal's actual body temperature. The thermometer on the left shows its preferred temperature or set point. Each type of animal has its own set point, a normal or target value, or each regulated internal condition. The set point is like the target value on a thermostat.
So one thing I want to quickly point out is they're using temperature as an example here. But every internal condition, the ones they talked about at the beginning, things like blood pressure, oxygen level, hydration, every one of those is going to use this same mechanism, this idea that there's a set point in ideal situations that the body tries to keep their internal environment at. So temperature is just the example here because sometimes it's sort of the easiest one to understand.
Sensors are cells, organs, or structures located throughout the body that perceive changes in some parameter of the external or internal environment. An example is body temperature. In animals, sensors are nerve cells specialized to detect a specific type of stimulus. Sensors continuously transmit information from this stimulus to an integrator. An integrator is a component of an animal's nervous system that compares the incoming information with the set point. Most integrators are a part of the brain or central nervous system. If the value of the stimulus being sensed is higher or lower than the set point, the integrator sends this information to an effector, a cell, organ, or structure that takes appropriate action to return the value to the set point. The process of homeostatic control is continuous, so sensors also monitor changes made by the effector. Consider what happens in a mammal when the sensor detects a body temperature that is lower than the set point. Sensory information flows from the sensor to the integrator. The integrator then compares this information with the set point and detects that body temperature is too low. Next, the integrator sends instructions to the effector. The effector responds by taking action, for example, inducing shivering to generate more metabolic heat, or fluffing up fur to improve insulation, to move body temperature back toward the set point. The sensor measures the modified temperature and sends this information to the integrator. This mechanism continues until the set point has been reached. When the body temperature overshoots the set point, the integrator detects that the temperature is too high and sends instructions to the effector. The effector responds with actions to increase the removal of heat from the body. These actions might include inducing panting and dilating blood vessels. The sensor detects the new body temperature and sends the information to the integrator. When body temperature is back to the set point, the integrator stops signaling the effector to take action. In this way, any deviation from the set point initiates metabolic and behavioral changes to achieve homeostasis. Homeostatic systems are based on negative feedback, a corrective response in which effectors reduce or oppose the change in internal conditions.
So this idea of negative feedback is when the sensor detects a difference that the integrator indicates is wrong, is not correct. It's not at this sort of set point, not this ideal situation. The signal that is sent is going to try and move it towards this optimal condition. The negative feedback is countering what the sensor is receiving. It's trying to move it away from what the sensor is receiving, either because it's too above the set point or below the set point. Positive feedback is a different system entirely. That's when you would be receiving input that the information is changing and you're trying to speed up the change. But the system of homeostasis is based on this negative feedback. So you can try and sort of keep every sensor, every system at sort of this optimal level. So when we think about this homeostasis, the idea is that you have this animal body. This is a beautiful representation of an animal body. I think I could actually draw that. But with this animal body, you have an internal environment and an external environment. Externally, there's going to be a lot of changes going on. As we said, the easiest one to be thinking about is sort of the idea of temperature. Temperature changes drastically on a daily basis and on an hourly basis. But within the animal body itself, you want to have as few changes occurring as possible. Now, there are some cases where you don't have animals worrying about temperature. What are those going to be? What kind of animals aren't going to have homeostatic mechanisms for controlling temperature? Ectotherms. They're going to be absorbed. They're going to be getting their temperature from the outside environment. They can't engage in metabolic activity. Now, having said that, they still have an optimal temperature. That's why they sit and bask out in the sun. That is what their effector is. That's what they're doing to change their internal temperature. They can't change it by doing something like shivering. So they sit out in the sun, and what happens when their temperature gets too warm, when their sensors are saying, oh, we're above the optimal temperature now? If you have a snake or a lizard sitting out on a rock in the middle of the sun, what's it going to do? Move away, go in the shade, protect itself a little bit. So that still is a homeostatic mechanism. It's just a little bit not as fine-tuned as we see with endotherms that need to maintain a constant body temperature. Yes?
So like what about when they get sick? I know I read in the book that sometimes they want to induce themselves fever, like we get fever so that...
Well, a fever is an immune response to an invading pathogen.
Yeah, because I know some of the cold-blooded though, they purposely lay out in the sun longer than they're supposed to so the body temperature could actually increase above the normal temperature so they could cure those pathogens.
Yeah, I mean, basically the idea of a fever is when you're ill, you raise the body temperature above what the pathogen can survive. You're trying to kill the bacteria or the virus by cooking it. That's what we do that's why we get fevers, because we're trying to raise our body temperature, and so that the virus or the pathogen, the bacteria is going to die. Same kind of thing there. The problem is if you raise it too much, you're not only cooking the pathogen, you're cooking yourself. So when we have this homeostatic mechanism, we're really trying to talk about maintaining small internal fluctuations. So the internal environment, the internal chemistry, the internal temperature is not going to vary very much. So as we said, this is done through negative feedback mechanisms. When you have changing conditions, these are detected by the sensors. Sensors can be cells, they can be membrane receptors. So you can have a sensor that detects temperature, or you can have a receptor inside the membrane of a cell that might detect things like oxygen levels or blood pressure. So there are a lot of different kinds of ways that you can have sensors receiving the input. Some of it is going to be based on the external conditions, but some of it is also going to be based on internal conditions. The information that the sensor receives is then sent to the integrating center. This is usually the brain, the spinal cord, but in some cases it's just an endocrine gland that is then going to release some sort of hormone in response to this sensory input. The integrators are sometimes called the comparators because they're simply comparing what the actual value is to what this sort of ideal set point is. If it's not within the tolerance range, if it's too high or too low, then what happens is the integrating center is going to send information to an effector. The effector is what is going to actually be able to change the condition. It can be changed either through chemical means, it can be changed through metabolic means, it can be changed through behavioral means. So there's a lot of different ways that you can respond when conditions are not ideal. Depending on what the condition is, the body is going to have a set series of responses. They mentioned a couple of responses if the temperature is too low. What are some of those?
Shivering.
Shivering. That's a behavioral response. What does shivering do? Well, make your muscles vibrate, move back and forth very quickly. You're using up a lot of energy. It increases the heat. So that's going to increase the body temperature if the temperature is too low. What was another one?
The fur.
Increasing the insulation, puffing the fur up. We talked a little bit about this before. But when the fur expands, it increases the insulation. It decreases the amount of heat that is lost. So while it's not directly increasing the heat, it's preventing heat from being lost. You retain more heat inside. The body temperature goes up. Now, what if your temperature was too high? They mentioned a couple things that can occur that way. What are some of those? Sweating. That is one. They didn't mention it, though. But sweating, when you start sweating, the body evaporates. The process of evaporation removes heat from the skin, from the body itself, and cools the body down. They mentioned things like dilating blood vessels near the surface. Blood stores and holds a lot of heat. But when it gets close to the surface, what happens to that heat? It gets lost. It's radiated out. If someone is really flush and you put their hand on their cheek, or if they have a really sweaty and red arms from flush skin, and you put your hand on their arm, what does it feel like? Besides maybe wet from the sweat. What does it feel like? Hot, cold? Warm. It's because all that heat is radiating out from the body. So when the body temperature is too high, that's one of the ways that the body can lose heat. By bringing blood to the surface, and then that heat is lost. So those are examples of how the effector can control what is happening to the temperature. So the idea is the effector then changes the conditions that are currently too far away from the set point towards what the set point needs to be. So again, we sort of have this idea, we have the set point. If there's a sensor that's detecting you're away from the set point, an internal or an external stimulus, something outside the sensor is saying we're not at what this set point is, it will then send that signal to the integrator. The integrator sends the signal, or sends the information that the sensor sends and says, oh, we need to compare it. We need to see how far we need to move. Are we at the set point, or do we need to change somehow? If you need to change, if you're away from the set point, because that arrow is too large, then the integrator will send a signal to the effector saying, do this. We need to change this condition. We need to increase the temperature or decrease the temperature or increase blood pressure. And so then the effector will simply do it. That will be some sort of internal or external response. In this case, the temperature will change, and then the sensor detects that new temperature. So this is a continual process. The sensor is always detecting what all these conditions are like. It's always sending that information to the integrator. The integrator is always comparing what the actual conditions are to what the set point is. And if it needs to be changed, it then sends that information to the effector. Now, with a body temperature homeostasis, as I say, it's sort of easy to wrap your mind around this one. But when you have this situation, you have the sensors. They can be detecting external temperatures of heat or cold. Temperature receptors would include things like the skin. You actually have receptors on your spinal cord. You have receptors on the hypothalamus. So there are a number of temperature receptors throughout the body, not just on the skin. But whatever information they receive, they send that to the temperature control center. This is in the hypothalamus. Different integrators are going to be found in different parts of the brain or different parts of the central nervous system. In the hypothalamus, they're looking at temperature. The integrator is going to be saying, is the body temperature above or below the set point? It sends that feedback or that information to the effectors. The response is going to depend, the effector that's going to be used is going to depend on what the integrator is sending. If the temperature is too high, the body temperature is above the set point, the effectors might do things like they're going to do things to decrease the loss of blood, or increase the loss of heat. So they might dilate the blood vessels near the surface. This is going to release heat. They're going to stimulate the sweat glands. That's going to cool the body down. They're going to influence the respiratory centers. Breathing heavy is another way that you lose heat. So every time your body temperature is too high, these are going to be the automatic responses that the body's going to engage in to try and lose some of that heat. If the body's temperature is below the set point, and they need to increase the heat, you're going to do some of these things where you constrict the blood vessels. If dilating them loses heat, constricting them is going to try and conserve heat. The process of shivering is going to generate heat to try and increase the body temperature. And then, as we said, you also might be doing things like increasing the fur for insulation. There are also going to be things that are going to occur at the respiration center, the heart center, to try and increase or decrease the panting or heavy breathing, increase the heart rates, and so forth. As all this is occurring, the sensor is continually monitoring what's happening. It's going to be continually adjusting what the signals are being sent are. Now, we have set points for every single facet of biological requirements. Temperature, blood glucose concentration, ion concentration. Every single ion that you can think that we might need in the body is going to have a specific set point. Tension within the tendons has a set point. Everything that you can think of that is going to have an ideal situation is going to be controlled through this homeostatic mechanism. The integrating centers are usually part of the brain and the spinal cord. The sensors are going to be found throughout the body, internal and externally. The integrating centers are in the brain and the spinal cord. The effectors are usually some sort of muscles or glands. Muscles can actually cause a physical motion. The glands can oftentimes produce sort of biochemical changes. And that brings us to the end of chapter 43. We now move on to start talking about what this actual nervous system is. What's actually going on with these nerve cells when they're transmitting signals. Now, as you sort of understand now, this nervous system is absolutely essential. All animals need to be able to detect stimuli. They need to be able to receive external and internal cues about the environment. This is done through the nervous system. But not only do they need to be able to detect them, they also need to be able to respond to them. And the way that you respond, that information is sent through the nervous system. The response usually isn't the nervous system itself, but the nervous system controls the signals that are being sent around. So when you have these sensory receptors receiving information, those are going to be detecting stimuli. You have motor effectors. These are going to be responding to stimuli. And then you also have the nervous system that links the two. The sensors are receiving the information. The motory receptors are responding to the information. But then you have the neurons that have to connect the two. Now, vertebrates themselves have three classes of neurons. They have the sensory neurons. These are sometimes referred to as the afferent neurons. These are going to be ones that are carrying impulses, carrying signals to the central nervous system. You also have motor neurons. These are sometimes referred to as efferent neurons. These are the ones that are going to be carrying signals away from the central nervous system to what is actually going to be the effector. And then in between, you have the interneurons, sometimes referred to as association neurons. Interneurons are going to provide the complex reflex and associative functions. Thinking, brain behavior, conception, those are all aspects of interneuron behavior, interneuron possibility. So the interneurons are found in between these sensory and motor neurons. Now, as we said, there's sort of two components of the nervous system. You have the central nervous system that consists of the brain and the spinal cord. You have the peripheral nervous system that consists of these sensory and motor neurons. The peripheral nervous system is broken down into both the somatic nervous system, so it's the somatic peripheral nervous system. This is responsible for controlling skeletal muscles. What does that mean about the somatic nervous system? Is it under your control or not? It's voluntary. You then have the autonomic peripheral nervous system. These are the ones that are going to be responsible for controlling the muscles that are found in the smooth and cardiac muscles. These are also going to be responsible for sending signals to the gland. Is this under your control? No, automatic. That's where it gets the name, autonomic peripheral nervous system. So one of these you can consciously control, one of them you cannot. So if you sort of look at what's going on with this whole thing is the nervous system, you have a peripheral nervous system component that's receiving information, the senses. Those nerve cells are then sending that information to the central nervous system. Now in this case, the brain is one cell. It is not really like that, but this is the central nervous system comprised of the interneurons. It interprets that information, then sends a response to the motor neurons that are then again part of the peripheral nervous system. So this is basically what your nervous system is. It's a little more complex than that, but overall that's the general idea. You can also think about it, what's going on in each of these areas. So the peripheral nervous system is receiving sensory input, potentially from an internal environment or from the external environment. There are specific pathways that are going to receive specific kinds of information. That information is then sent to the central nervous system where it's processed. A signal is then sent to the motor pathways. It's either sent to the somatic nervous system, which is under voluntary control. These are going to be the skeletal muscles. Or it's going to be sent to the autonomic nervous system, which is in involuntary control. The autonomic nervous system is then broken down into two further components. You have what's referred to as the sympathetic nervous system or the parasympathetic nervous system. These are both involuntary responses. The sympathetic nervous system is referred to as the fight or flight response. It doesn't necessarily mean you're going to start fighting somebody, or it doesn't necessarily mean you're going to start running away. What it means is those are biochemical reactions that occur in situations where fight or flight are their appropriate responses. These things can include things like dilating or constricting the pupils, increasing the release of adrenaline. Those are all parts of the sympathetic nervous system. The parasympathetic nervous system is responsible for controlling the rest and repose responses. So if one of those, for example, is going to increase the flow of adrenaline, the parasympathetic is going to decrease the flow of adrenaline. Anyone ever been too hyped up to sleep or too excited about something? Maybe you saw a really good TV show or something like that. You're all, oh, that's so awesome. I'm so psyched about it. That's an overactive sympathetic nervous system. Especially if it's about 12:30 in the morning, you're thinking, oh, I just want to sleep. In that case, you have to be telling yourself, why is my parasympathetic nervous system not functioning correctly? So with this nervous system, all the neurons that are found in this nervous system have the same basic structure. They have the cell body, which we talked about. This is the enlarged part of the cell itself that contains the cell nucleus. They have the dendrites, which are the short cytoplasmic extensions. These are responsible for receiving stimuli. They can either receive stimuli from the sensory organ itself, like the tongue or like the skin, or they can receive stimuli from other nerve cells as the signal is being transmitted along. And then you have the axon, which is a long extension. There's usually only one axon, and it extends well past the dendrites, and it is responsible for conducting the nerve signal away from the cell body to either the next nerve cell or to whatever motor effector is going to be receiving the signal. So regardless of whether we're talking about sensory neurons, motor neurons, or interneurons, they're all going to have the same basic structure. This is the structure of the nerve cell. You have the cell body, you have the dendrites that receive the signal, either from other nerve cells or from the environment. The signal is then conducted away along the axon. Now, the way that a nerve signal works is it's all about electrical gradients, electrical potential differential. There's an electrical charge difference between on the inside of the nerve cell and on the outside of the nerve cell. So there's actually a chemical difference, an electrical difference in every single nerve cell's plasma membrane. And this is the basic function, the basic idea of how nerve cells work. You have a negative side. This is the cytoplasmic side inside the cell. is going to be more negative, and you have a positive side. The extracellular side outside the cell is going to be more positive. So when you have this electrical charge difference, the way a nerve signal works is basically by moving that electrical charge. So when a neuron is not being stimulated, you still have this positive and negative. It's what's called the resting potential, the charge difference between the inside and the outside of the cell. It ranges on the cell types from anywhere from -40 to -90 millivolts. It's a negative number because the outside is positive, the inside is negative. So that's just the way that we measure it. So there's this resting potential. And when that resting potential changes, when you actually change it so that it becomes positive on the inside and negative on the outside, that is what a nerve signal is. That is what a nerve impulse is. So the inside of the cell is more negatively charged than the outside. The way that the charges change is through pumps and channels. When we talked about cells, when we talked about membrane proteins, we talked about things like the pumps and the channels that allow ions to pass through. What are ions? They're molecules that have a what? A charge. They're either going to be positively charged or negatively charged. If you're moving an ion through the plasma membrane, that is going to influence the charge that is already existing with this resting potential. So, for example, the sodium-potassium pump is one of the pumps that is responsible for helping to change this potential. What the sodium-potassium pump does is it brings in 2 positive potassium ions for every time that it pumps out 3 positive sodium ions. So what is that doing to the charge? More negative on the inside? Well, it's actually making it more positive on the outside. Sort of the same idea, but it's a little bit different because you're not actually moving negative ions. The only thing you're moving here are positive ions. You're moving 2 positive ions in, 3 positive ions out. You're increasing the positive charge on the outside. Another example is what's called the ion leakage channel. Ion leakage channel really specifically also works with potassium and sodium, but what it does is it doesn't actively pump anything. It basically allows potassium to diffuse out at a faster rate than sodium diffuses in. So you're moving more potassium out than sodium in. What does this do to the charge?
It's going to make it more positive.
More positive on the outside. Same thing. Doesn't matter if it's different kinds of molecules. All you're doing is looking at what the ion charge is. If it's positive and there's more positives moving in one direction, that's going to produce a greater positive charge. So all of these are going to be increasing the positive. These are both going to be increasing the positive charge on the outside, decreasing the positive charge on the inside. So sort of creating this resting potential. Not all channels and pumps do this kind of behavior. There are some that are responsible for moving negative ions. There are some that are going to be responsible for moving more positive ions into the cell. But the sodium-potassium pump, this one that is increasing the sodium on the outside relative to the potassium on the inside, is the primary pump that is responsible for creating this resting potential. So what you end up with a situation where you have a very high concentration of sodium ions outside the cell relative to inside the cell. You're going to have a higher positive charge on the outside. This is what the resting potential is. This is what creates the resting potential. This is what's going to occur in a normal situation. Now the membrane, just like it's not permeable to positive ions, is also not permeable to negative ions. Because you have a greater positive charge on the outside, through this pump action, you also end up with a greater negative charge on the inside. So you have this differential, and this is how the resting potential is created. If you look at the cell, you have the positive on the outside, you have the negative on the inside, you have this pump that's moving things around. This is a channel that allows ions to flow through, but ultimately you're going to have more negative ions on the inside, more positive ions on the outside, creating this resting potential. Once the resting potential has been established, and it's pretty much always going to be present, and once you have this resting potential, then you can conduct a nerve signal. So what actually is the nerve signal? What is the nerve impulse itself? It is what occurs when you change this ion concentration, when you change the charge that's associated with these cells.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge.
That's over here, right there.
This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside of the cell.
So that's what a nerve signal is. Every time you're receiving an input, a sensory input, or deciding to do something, to move a body part, this is how this signal is being sent along. You have this resting potential. You then have a depolarization where you're changing the positive outside and negative inside to sort of flipping it around. This is what the signal is as it's being sent along the nerve cell. You then have to reestablish the resting potential so the cell can be used again. That's called a repolarization. So the depolarization brings the positives inside, so it changes the positive and negative, so the positives are now inside the cell. The repolarization moves it back to what the resting potential is. And you can start to imagine how quickly this occurs. If I were to take somebody's hand and prick them with a needle, what is their response going to be? Hopefully they don't just say, ow. I'm hoping they jerk their hand away or something like that. If you put your hand on a hot stove, you jerk it away. So you can get an idea how fast this depolarization and repolarization can take place. It almost seems like it's instantaneous. It is not. You can measure the time it takes. But it is so fast, that is how nerve responses occur. That is how we're able to respond to things both at a conscious level and an unconscious or subconscious level. So we'll continue talking about nerves on Monday, talking about the two different kinds of changes. Can everyone please sit down until I'm finished talking? We will continue talking about the nerves on Monday, talking about two kinds of changes. They mentioned action potential in the animation. That's an all or nothing response. We also have what's referred to as a graded potential, which is sort of a continuous response. I will see you all on Monday.